I'm a lab manager from the Department of Physics from the University of Haifa, from the Lab for Biological Physics of Cells. And some of the things we do as a lab for biological physics is basically model biological processes using non-living matter. So the beautiful phenomenon we studied is propagation of phase boundary in polyacrylate gels, which can be related to electrical pulse propagation in nerve cells. So sorry for being repetitive. A brief introduction about hydrogels, since polyacrylates are hydrogels. The hydrogels are a 3D network that contains large amounts of water. They can be cross-linked either chemically or physically. And they have highly hydrophilic groups along their chains, what makes them swell when they immerse in water. They have very interesting properties, and some can undergo volume phase transitions as a response to all kinds of stimulation as pH, solvent quality, temperature, mechanical stress, ionic contact, and so on. So this is an example for a thermoresponsive polymer, NIPAM copolymerized with HEMA, which you can see a gradual decrease with temperature until we get to a certain point when we see a steep decrease in the volume of the polymer. It loses its volume. This is the phase transition, a volume transition. And the second example is the polymer we worked with, sodium polyacrylate. When we put it in a equilibrium solution of sodium chloride 40 millimolar, and then adding to that a divalent ion such as calcium, we see a gradual release, a decrease of the volume of the gel with adding the calcium ions until we get to the critical point, to the critical concentration when the polymer loses expels water and loses its volume. As you heard in this workshop, volume transition can play a major role in biological processes, such as DNA compaction, membraneless organelles, and so on. And one of the examples, a very famous biological hydrogel is the mucus gel. That its mechanism is related, the swelling and the deswelling in the cellular system is related to D and monovalent ion exchange, but in this case, this is a solvent quality example. When we get to a composition of 50% water and 50% glycerol, the gel collapses and loses its volume. This is probably a plot that I want to convince you why polyacrylates are a good model for biopolymers. You can see in the plot in the blue line DNA gel, and in the red line polyacrylate, sodium polyacrylate, and it reacts to divalent ions. And we see that in the critical concentration of strontium or calcium, we can see a steep decrease in the volume, also for polyacrylate gels and also for DNA gels. So a bit about sodium polyacrylate. You can see, you can see here the repeating unit of sodium polyacrylate, the carboxylate group, and the counter ion sodium. It can be cross-linked using bis-methylacrylamide. It has the carboxylate group, but with the pH, it can be protonated and become carboxylic group. So the network in the right figure can be seen, the crosslinks, the carboxylic acid groups and sodium carboxylate groups. We can immerse it in water and then let the gel swell due to the hydrophilic groups and also due to the osmotic pressure. About volume transition due to ionic contact change in sodium polyacrylate, the monovalent ions in the left column, in the left figure, can be seen that we get a gradual decrease in the volume as we add more salt. But the degree of swelling, by the way, it's the ratio between the dry and the wet gel, and it's approximately for lightly cross-linked gels, the mass divided by the mass of the wet gel divided by the mass of the dry gel. So as we add monovalent salt, we can see that we have a gradual decrease, and there's no significant change in the identity of the monovalent cation. However, for divalent ions, in the second group of the periodic table, there is a decrease in the degree of swelling until we get to the critical point, and then we can see the steep change. And in this case, we have a dependency on the identity of the ion. As we go down in the second column of the periodic table, we can see that we need less concentration of the divalent cation. And some of the reasons for that can be that as we go down in the table, the hydration shell becomes weaker. And also, as we go down the table, the nature of the bond, the chemical bond that bridges between two carboxylate groups, becomes more covalent, which is stronger than the ionic nature of the upper elements in this column. And here in this picture, you can see a gel right before the collapse and right after the collapse of the gel. So we were inspired by Tasaki's work. Tasaki was a Japanese biophysicist and medical doctor. He studied brain cells, and he wanted to use sodium polyacrylates as a model for his phase transition paradigm in nerve cells. For that, he used sodium polyacrylate, and he immersed to mimic the geometry of the nerve cell. He used cylindrical sodium polyacrylates, and when one edge was immersed in a calcium chloride solution, 27 millimolar, and the second edge was immersed in 40 millimolar of sodium chloride. This is the equilibrium solution that was this gel were immersed overnight before the experiment. So he applied electrical current, and he saw that there is formation of phase transition, of phase boundary. You can see marked with the arrows here, and it was propagating in a velocity of 1 millimeter per 10 minutes. So he used the snapshots to calculate the velocity. And as we went further in this study, this very impressive work and phenomenon, we saw that there are things that needs to study further. Tasaki did all these experiments when he was 92 years old, which is quite impressive. But it lacked some details and understanding about this phenomenon. So what we wanted to see is understand about additional parameters. Tasaki used only one voltage value. He used only one gel diameter, though in his material and method section, we saw that he reported 2 gel diameters. In the results, we saw only one. He used only one concentration, this magic concentration. It is magic concentration of calcium chloride. And also his analysis, it wasn't, we wanted to perform more detailed analysis. So we used time-lapse videos of the gel. So this is how the experimental setup we used looks like. We used a 3D printed two-well chamber. Actually, it's a 4-well chamber, but this was for our convenience. It was equipped with reversible chloride electrodes, where one edge was immersed in a sodium chloride 40 millimolar solution, and the second edge was immersed in calcium-containing solution. We changed the concentrations of the calcium, and we applied several voltage values, and we put that system under a dissecting microscope to take the time-lapse movies. The time-lapse movie you're going to see now, each frame was taken in an interval of 5 seconds between each frame. And in real life, this movie is 75 minutes. So you can see that a phase boundary is forming and is propagating, and also the edge is propagating. Maybe I'll use that stick. And in the area exposed to air, we also can see that it shrinks down as the time goes by. The stripes, by the way, it's because of the 3D printing method. It prints in layers of 25 micrometers, which can also be good for calibration. We wanted to do a detailed analysis of this phenomenon. And we saw 3 observed deformation. These 3 frames from different times. When you can see, now use the stick. You can see that as times go by, this edge is moving. And also the phase boundary, the tail is elongated, the phase boundary is propagating, and also the area exposed to air gets thinner with time. The bath you see is the calcium bath, of course. And you see, I don't know if you can see, but there's the silver chloride electrode. So the velocities are Vm for middle, Ve for edge, and Vpb for phase boundary. And the analysis was done using object detection algorithm that was enveloping a rectangle around the collapsed phase of the gel. And we got the output was the coordinates of the phase boundary, the collapsed phase. And this was done by this very talented young man, a bachelor project student. He worked on this object detection algorithm, and he trained over 1,300 frames manually on the collapsed part of, he trained a neural network. So the results we got for the elongation of the tail of the phase boundary for 61 experiments. Out of these 61 experiments, 95% of them we got a linear response with time for the first 10 minutes. But after that, we saw 3 different behaviors. It either was a total linear response, or a square root response, as you can see in the middle, or a linear response with a halt. So after a certain time, the phase boundary was stopped from propagating. The phase boundary velocity was in the range of 0.2 to 1.7 micrometer per second, which was in the range Tasaki found in his experiments. In his experiments, it was 1.67 micrometer per second, converting his units he used. So we wanted to see how calcium concentration affecting the phase boundary propagation, and also how much time it takes for the tail to be formed. So we saw that as we increased the calcium concentration, the range was 10 to 27 millimolar. We saw that as we increased the calcium concentration, the velocity is increased monotonically, however, with a large variability. And also the tail formation, the tail was formed faster for the higher calcium concentration, and in a calcium concentration of 6 millimolar, we didn't see a phase boundary at all. So it was like kind of a threshold. That. We wanted to see how external voltage values affecting the phase boundary propagation. As we increased the voltage value, we saw that there is an increase in the calcium in the phase boundary velocity, which makes sense because we are accelerating the calcium ions into the gel. And by the way, Tasaki only used experiments with applying voltage, but we found out that we don't actually need this is spontaneous. Also with without applying an electrical voltage, there is a phase boundary formation. And as for the initiation time of the tail formation, we saw that the tail formation is being formed faster as we increase the voltage. We also wanted to check different gel diameters. The diameters you see here are the dry gel diameters. For these experiments, we used external voltage because we saw that for the larger gels, there is no boundary formation if we didn't apply electrical current. This was done with 0.15 volts. And as we increase, as the gel diameter is increased, the initiation time takes longer. And the velocity is also lower as we increase the gel diameter. Summarize. We saw three of the deformation of the gel. We saw there is shrinking in the middle. And I want to say something about this shrinkage of the area exposed to air. At the beginning, we saw that, and we were concerned that it affects the phase boundary propagation, or the edge actually propagation, and we wanted to verify it doesn't. So first, we want to verify that it's because of water evaporation. So we conducted two control experiments, and we saw, and putting a droplet in this area exposed to air of sodium chloride, the equilibrium solution, showed that this area doesn't shrink. So it's due to evaporation of water and not related to the calcium cations. And also the second control experiment is to see that the edge doesn't move faster because of this shrinkage. So we put in the two wells 40 millimolar sodium chloride, and we saw that both edges were not moving towards each other, although this area was shrinking. The two other deformations we saw is the V edge and the V phase boundary, of course. And we saw three different types of time dependencies of the tail elongation. One was completely linear, one was fitting better square root dependence, and one was linear with halt. we actually, if someone is interested, we would like to get a collaboration in modeling this phenomenon, because the work that we know, at least, are less are not fitting our parameters. There are two works that are relevant, but though one of them considered, they both consider different conditions. So if someone wants to collaborate, that would be great. We saw that as we increase the calcium concentration, the external voltage, or decreasing the gel diameter, the phase boundary velocity is getting higher, and the tail formation initiation time is getting lower. The high variability that you saw is probably due to some intrinsic factors differences between each gel we use, though we try to use the same conditions for each experiment. But there are some differences, such as the cross-linking density, or the fraction of charge between each gel, the distance from the electrodes, the electrical field. Somehow, of course, it affects, and we try to keep everything constant, but we got high variability. So thank you for listening, and this is some of the cool stuff we do in our lab related directly or indirectly to charged soft matter, action potential in giant algae, measuring sound wave in lipid monomers using Langmuir trough and FRET system, and chemical oscillators. So I want to thank the people that were involved in this work, Matan, the principal investigator, Dimitri, that did the object detection analysis, and also for our collaborators, Gary and Susan, his former talented student, and our group members for our great spirit we have in the group, and of course for the funding. Thank you very much. Yes.
In terms of fundamental.
Yes.
The fact that gels collapse
yes.
for a long time. Of course,
motivation.
More general.
Yes. So I mentioned Tasaki, and he studied brain cells. He used squid giant axons, and he saw that there is a phase, a volume transition in the polymer, the protein layer attached to the axon membrane. And he changed the concentration of calcium and sodium ions, and he saw that there is a mechanical change, changing the volume as he changes the concentration, and the excited state of the cell is related to the collapse state, to the swell state, to the sodium bound ions. And the collapse state is the resting state of the axon. So he wanted to use this polyacrylate gels as a charged soft matter, as these proteins in the axon membrane, to model this system. He was very excited about that, to find out that there is a synthetic polymer that can act as a model system for his paradigm. Yes. We basically we do all kinds of stuff that we try to mimic biological processes. So we are really interested in action potentials and how it's propagating through the cell, and also the picture that I show of the giant algae, the memory, when we put electrical current, we have a wave, a density wave, and you can see that. No, we haven't checked that, but I believe that if we will cross-link, from my estimation, if the cross-linking density will be higher, maybe it will be the collapse phase and the swell phase will be it will be less astonished because it will get harder to see the changes. Good question. It seems like it's homogeneous from the microscope, though we didn't use, this is a stereo microscope, it's not a high resolution microscope. It seems to be homogeneous, but yeah. But the collapse part, it fits the collapse values in the literature, the swelling degree and everything, so.
All the speakers